Welcome, everyone. Thank you for joining us for this edition of the GOA-ON webinar series. My name is Kalina Grab, and I am a GOA-ON Secretariat and a Knaus Fellow in the NOAA Ocean Acidification Program. And I'll help to introduce the moderator today for this webinar series. So this webinar series has 4 sponsoring organizations. First, GOA-ON, which is the Global Ocean Acidification Observing Network. Second, NOAA, which is the National Oceanic and Atmospheric Administration. Third, the IEA-OA-ICC, which is the International Atomic Energy Agency, Ocean Acidification International Coordination Center. And the IOC UNESCO, the Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific, and Cultural Organization. For those that are new to GOA-ON, it is a collaborative international network of over 1,000 members from 105 countries that is designed to detect and understand the drivers of ocean acidification and the resulting impacts on marine ecosystems. During the presentation, all participants are in listen-only mode. You're welcome to type any questions into the question box, which can be found on the bottom of the control panel on the right-hand side of your screen. We'll be moderating the incoming questions, and we'll pose them to our speakers during the question and answer section, which will be immediately following the last presentation of today. And today we have 2 presentations. So at this time, I would like to introduce our moderator, Dr. Carla Berghoff. Dr. Berghoff is the LAOCA co-chair. She is a researcher in the dynamics of marine plankton and climate change program of INIDEP, which aims to study long-term changes in the physical, chemical, and biological environment in the ecological time series stations EPEA, El Barril, and Costal located in the northern Argentine continental shelf. The focus of her research is to relate the temporal variability of the carbonate system and the role of phytoplankton on the regulation of air-sea gas exchange in the context of ocean acidification in different areas of the Argentine Sea, especially in the series of time with EPEA, El Barril, and Costal. So Carla, I'll turn it over to you.
Hi, thank you, Kalina. So today we have the honor to have Michelle and Thiago, and I'm gonna present Michelle now. She will be, she's a biologist with a PhD in chemical oceanography from the University of Concepción, Chile, and Paris, France. She is the head of chemical and geology laboratories at the Instituto del Mar de Peru, IMARPE, developing research associated with the Peruvian upwelling system, and in particular the oxygen minimum zones, nitrogen and carbon cycles, carbonate system acidification, and recently developing sensors for coastal marine stressors monitoring. She participates in the Latin American and the Caribbean REMARCO research network on marine coastal stressors and in different national and international projects. She was the former LAOCA co-chair, and she will speak today about the biogeochemistry of the Peruvian upwelling system in the changing ocean. So I'm gonna pass to Michelle.
Thank you, Carla. I will put the presentation. Okay. Okay. Well, good morning and good afternoon. I'm so happy to stay here. It's an honor. Thank you, Carla, and thank you for the GOA-ON and the LAOCA to organize this webinar. And it's exciting to present some of the results from the Peruvian system to all the acidification community. Well, first of all, I would like to present the team that worked with me and make and support all this research activity and the result that today I will present. This is a team with people from IMARPE, but also for other universities that we make some collaboration in projects like la Universidad Científica del Sur, and in the north of Peru, la Universidad Pedro Ruiz Gallo. And we also have some international collaborations, in particular with the Dr. David Point, and with some civil organization like Pro Acuicultura, with the biólogo Carlos Sotomayor. Well, and here Peru one of the country. And today, the idea in this presentation is first, make a short review about the characteristic of our system. Then look some past research on pH and other variables, and the present local monitoring efforts now and the initiative and project. And finally, looking about the report of the SDG and discuss some gaps, challenges, and opportunities for this kind of research in Latin America and in general in the region and the global. Okay, all the community recognize that the Humboldt upwelling system, and in particularly the northern component of the Humboldt system, that is the Peruvian upwelling system, is characterized by two peculiarities or conditions. In one way, the low pH, and also a very low oxygen conditions. And when we look the map and these figures, we can observe that, okay, this is a particularly hot spot that make that many research at international and national level are focused to understand how mechanisms regulate these conditions. These conditions are part of a complexity in a system that is regulated at different scales, in a temporal scales and spatial scales. We can have in the Pacific waters the signature of the large scale circulation. We have the oldest water masses, and this condition make that the chemistry of these waters contain very high nutrients, very high CO2, low oxygen, and low pH. And this is associated with the physical and the biogeochemistry of the organic matter remineralization. And at subsurface occur this whole process that because of a big physical process that is the upwelling process, these subsurface waters come into the surface and bring these high nutrients and the possibility and a favorable environment to develop the high productivity that characterize Peru, high primary productivity, and then these high fisheries like anchovy that are the most representative of our country. And the physics plus the chemistry finally turn in this condition of oxygen depletion and low pH that is that start by the physical process, but then is modulated and intensified by these biogeochemistry processes like productivity and organic matter remineralization. And here I put only another condition that appears and is very explosive related with the oxygen depletion, that is this event that we have and several times in the year in some close bays, but also in other places along the coast of Peru, that is the sulfur plumes, that is a consequence, a direct consequence of the productivity and the organic matter remineralization. And this condition is also connected with the pH condition and finally all the biology and ecology. Okay, we have a very productive, naturally eutrophic system, sour and breathless. And the other condition that is very important and is a challenge for us is all these characteristics are in a very, very high variable system. Variable in temporal and in a spatial condition. Here I present some result of two publications that we performed in Peru. One is from Chavez that show, for example, the seasonal condition of the chlorophyll A, indicator of productivity, the winds, that is the driver of the upwelling and the insolation. And we can look that we have productivity out of phase with the maximum upwelling forcing. This is one of the characteristics of our system. Winter, we have the most intensive upwelling, low productivity, and one hypothesis is related with a low irradiation in the system. Es decir, we have all the year an upwelling condition, but the productivity has this seasonality. And here, if we can look in this climatology that we perform for Callao, we can observe the change also in the temperature associated with the difference in the upwelling forcing. For example, here the maximum, and we call this condition. And this is also looking in the oxygen variability that we can observe and semi-annual signature enhanced, this is driven by the upwelling, but enhanced by the respiration during summer because of the higher productivity. But we don't have only this seasonal signature. We have also interannual signature, and everybody knows the big impact that we have related with El Niño. And the ENSO cycle, we have El Niño, La Niña, we have different types of El Niño, and a direct effect and impact over the chemistry conditions in the water columns, finally the productivity and the ecology of the system. Here we reconstruct a time series from IMARPE. IMARPE, since 50 years ago, more than 50 years ago, make a high effort to sustain and monitor the environmental conditions in the ocean, because this is very important for our biodiversity and the management of fisheries. And here in this time series, this is for the oxycline, that is a proxy of the change in the oxygen minimum zone and the habitat distribution. And here the chlorophyll A, and we can look in the shadow part, all the El Niño event that we have in the last decades. The big El Niño, like in 82, here 97, but then in the last decade, a very high frequency on this other El Niño costero and the El Niño canónico or Modoki. And we can observe that the interannual variability is very strong. We can say the big signature of the shoaling of the oxycline during non-El Niño period and the deepening, for example, during El Niño, that make very complex to perform and to identify attendance in the time series. And okay, this is interannual. We have seasonal, interannual, but we have also interseasonal, decadal, and at a spatial level, we have also several variability associated with different latitudes related with different conditions, all the tropical system, the equatorial system, and then the upwelling in the central and south part of Peru. And also a very important mesoscale activity that over the year that also made some changes in the biogeochemistry features and in general the system. Okay, we have a big challenge and we need to sustain ocean observation in order to track and to understand the biogeochemistry and the high variability of the system. What about the carbonate system? Well, we start, there are some, I would like to show some, I choose some results from the all and new publication related with some analysis associated with the carbonate system, PCO2 or pH or alkalinity, in order to show that we know something about the system. We have in the 80s and the 90s some studies related with international cruises that identify that the pH is very low in the system and is directly connected to the upwelling condition that determine this condition for Peru as a very low pH. And then probably the first work that established the role related with the CO2 for the Peruvian upwelling system was the work of Frederick Chávez, Ledesma, y colaboradores that determine in Peru the very high local CO2 production and a net source of CO2 to the atmosphere that is the signature in our system. There are some upwelling systems that can, by the pumping, biological pump, make a sequestration of the CO2. In our case, the physical pattern is very significant, and all the year we can observe a CO2 flux of CO2 to the atmosphere. And then the first local study related with the pH is the León, the Violeta León, and co-authors that perform a work based on 3 years observation. And in this publication that is from the IMARPE bulletin series, you can observe the connection and connectivity between the physical and the biological processes that modulate the pH. And finally, we have the recent work of Martina Hernandez Ayón, also in Peru, during a cruise, during a normal year, that show and confirm this low condition of pH related with the high PCO2 condition, and show that the system could be very vulnerable because of the very shallow condition of the pH, in particularly in the central area, that can affect the saturation of carbonate and calcita and aragonita, and probably impact in the organisms. Okay, and what is the present? The present is from the last year, we start different national and international collaboration in order to reinforce the capacity about the measurements of pH and all the carbonate system. This is something that we perform with REMARCO and also with the project of the IAEA, the RLA 7025. And in the other side, the idea to have a project that try to, in a multidisciplinary condition, study the Peruvian system and try to explore how perform observatories, environmental observatories to monitor the marine coastal area, in particularly stressors, but with the particularly to be the low-cost observatory for that. Because we understand that it's something very important to have a low-cost condition in our sensors and in our measurements, but in another side, we need low, but very, with a high precision in our analysis. Okay, and this is the last result. This is part, for example, of the master thesis and a publication that is working by Wilson Carwapoma, related with the condition, the carbonate system, and the Peruvian upwelling digamos, comparing the north to the south and different temporal conditions associated with neutral or normal condition and El Niño, in order to track more the variability and understand more what happened between the coastal and the oceanic area and between the north and the south in the condition of the pH, the processes, and in general the carbonate system. Here we can observe, for example, that in summer, this is the first figure, we can look in the warm colors, the very high levels of pH compared to for example, the very coastal measurements or the measurement that we will look during winter. This is modulated by the productivity and in particularly by the blooms of the hubs of the red tides blooms that we have several with a high frequency in all the coasts during the summer. And in the last time, during the summer, sometime in winter, and also in spring. Entonces, this change the condition of the pH compared with the winter. For example, this is a neutral winter, and this is a winter during El Niño. And we can observe that we have low values of pH, here the lowest values, and here you can observe, for example, the upwelling cells, the influence of the physical processes. And here we can observe a higher values of pH, and this is modulate by all the change in the water masses and the influence of El Niño that modify the influence of the upwelling, and several times the upwelling is reduced or in the system. And this you can observe here. This is a project that we started in 2013 until now. And here the salinity to look what is the change in the water masses. I only look that you look the green versus the blue. The green is the cold coastal water, that is the upwelling waters. During a normal El Niño, during a normal year, you have this condition, all coastal water condition, and very low pH that go to the oceanic part. And in the oceanic, you have high levels related with another water masses, that is the subtropical surface water, that during El Niño come into the coastal, modifying very significant the condition of the pH, that increasing this pH, and in particular affecting the ecology of the plankton that change, and also change all the carbonate, for example, planktonic organisms like theropods or coccolithophores that are modified because of this change in the water masses. But not only we have the difference in a temporal scale, we have also difference at latitudinal scale. And because we have some effort during some cruise to monitor between the coastal and the oceanic area, we can compare, for example, the north, like is Paita or Chicama, versus the central Callao and the central South Pisco, and looking what happened in the condition of temperature, oxygen, pH, and the saturation of aragonite. And this is very important because we need to, the variability is so high. In the north part, we can observe that the condition of oxygen are not so, the depletion is not so significant like in the central part and the south. We can observe that the pH is higher compared to the north, that is the Callao, for example, that is very shallow, the low pH condition, and also in the coastal area in the south, for example, and in the central area. And then, for example, we can look in terms of the habitat for some organisms, a different condition between the north, that is more wide and more not restricted in the central area. This could be something very important to monitor and know, because we can give this information for the policymakers and make some adaptation strategies to management the organisms. Okay, and related to the observatory, this is some very new data because we started in the pandemic this project to implement in San Lorenzo, Callao, a local observatory to monitor in a high resolution all the physical and some chemistry variables. Here I will present only the high resolution, no, without autonomous methods from temperature, oxygen, pH. And the interesting thing is this year we have a El Niño Costero, and now we are in a El Niño year. And with this very coastal observatory, we can track very well the changes, for example, in temperature and the signature of this El Niño in the coastal area, and then the change, for example, in the oxygen that made this tendency to increase temperature, oxygen, and pH. And we compare this, for example, with the wind. This is very important because the wind is the driver of the upwelling, and during El Niño, we can have, for example, in El Niño Costero, a very low winds in the system, the decrease in the velocities, and we can observe here, and it's directly related with the change in the physical and the chemistry of the system. And this is part of the monitoring that support also, for example, the monitoring of El Niño for Peru, and it's part of the result that sustain the prediction of El Niño. Well, and finally, we also work, and Wilson Kawapoma is one of the people that drive all the analysis of the carbonate system, and he was one of the first generation of the LAOCA training in 2014, I think. And then he continued, and now in the context of the REMARCO activities, we have the goal to report the SDG 1431, and we are now in the UNESCO. We start to report in December or in January 2023, and we are very enthusiastic because it's not only report a data from a fixed station, it's also to implement a measurement of the pH with a quality, a very high quality. And I think this is very important for the community. And also we have all an exercise to harmonize in the region the protocols in order to measure in the same way, and can report and have more countries in the Latin America and the Caribbean that can report this indicator of the SDG. Okay, and you can visit the page and you can look all the countries that now report the data. Okay, I think this is only to show that we have this, that we make the research in the coastal and oceanic from the Peruvian coast is also important in the bays. We have several bays with a high activity of aquaculture. This is two examples. One is from Lande Merma in Paracas that show the change, for example, in oxygen and pH, and relate this extreme anoxic event with very low pH condition. And this is very important for the aquaculture, the concha abanico in the area. And also we have some example that is from Natalie Bravo and Jorge Fupuyan and Wilmer Carvajal, that they started in 2017 in Sechura, and now they continue to perform these activities. See, we need to measure and understand at different levels, and the studies and work in the base are very important. And very important because we need to understand what is the future. And here I put two papers that is very interesting to read and to make the reflection. One is about the vulnerability of Peru related with the ocean acidification, and the other is related with the scenarios associated with the change, for example, in the saturation of calcite and aragonite, and finally the condition favorable or not from the environment for the different species. And for that, the only way to know the future and to understand more is continue to about the measurements, about the studies, experimental studies in the biology, and with data, we can manage better the future, and we can inform better and to be less vulnerable that present in these articles. And for that, I put some ideas related with gaps, challenge, and opportunities. One is related with the observations. We need to continue, we need to increase the observations in our countries, and these observations are very important for the experimental point of view, to experiment with organisms in a real conditions. And this is very important to know the tendency and to know finally what could be the conditions in future scenarios related with the pH, but not only the pH. The pH is only one variable, and we understand that we need to monitor together with other key variables, no? Like dissolved oxygen, the physical conditions, the nutrients, the chlorophyll A, because all is very articulate and integrate in this complex system. We need new technology. Yes, we need low-cost technology for the carbonate system. I think this is very important. Probably for the physical part is more developed for oxygen, but in the pH and carbonate system, I think we need to more innovation and more technology. And also we need to go to the stakeholders probably and try to include and look how we can monitor together, how we can assess the impact forecast related with the condition in the system. Then I think another part that is key is the modeling. We need to, the modeling offer the possibility to go to the future scenarios, but also to test some hypotheses about the process that modulate in a spatial and temporal change. And I think it's very important, no, to work more close with modelers. And finally, I don't put the biology, but okay, we need, no? The biology part is very important, low related with experimentation and observation of the organisms. But I put the final related with the networking and the international collaboration. I think it's so important, the articulation. We can be more stronger together and we can learn a lot working together, no? With the different organization, and the importance to monitoring and provide data for the international and national database. And one of these is the SDG. And I invite all the people to try to put in the laboratories the expertise to implement that and have more data to know more about the environment and the prediction for the future. And okay, thanks. I don't know if I good on time. And well, this is the team during the virtual condition and at the sea. And thank you very much for all the people that work in my team in Peru. And thank you also to the Prociencia, Concitec, the Instituto del Mar del Perú, por supuesto, that bring all the facilities to make this kind of research and continue to obtain data of quality for science and for improve the science in Peru. Thank you.
Thank you, Michelle, for this interesting talk. It was really nice. We are going to pass the torch to Thiago, and then we're gonna have some time for questions and answers. So now I'm gonna introduce Thiago. Thiago Monteiro is an oceanographer, a master and PhD in physical, chemical, and geological oceanography. Currently, he is a collaborator in the Carbon Team Research Group at the Laboratory for Oceans and Climate Study, LAOCA, FURG in Brazil, working with sea air fluxes, marine carbonate system, and biogeochemistry. He is a member of BrOA, which is the network for Brazilian for ocean acidification, and also a member of the MARES project, which aims to disseminate knowledge of oceanography and Antarctic studies to civil society in general. He's mainly interested in the integrated oceanographic processes. He will speak today about the biogeochemical studies in the tropical and South Atlantic Ocean, from air-CO2 exchanges to ocean acidification. So thank you. And now we are going to listen to Thiago. Thiago, you can unmute.
Okay, thank you, Carla. Can anybody see my screen?
Yeah, it's perfect.
Okay, thank you. So thank you very much, Carla, for this introduction. So as Carla said, I'm Thiago Monteiro from the Federal University of Rio Grande in Brazil. And it's a great pleasure to be here to talking about our studies on biogeochemical and in different regions of the Atlantic Ocean. So actually, I'm going to talk about some of our studies on the biogeochemistry in different regions and the studies carried out by the Brazilian Ocean Acidification Network. And so I'm going to talk about some studies from the tropical Atlantic to the northern Antarctic Peninsula, including some studies on the SE-CO2 exchange and the ocean acidification. So from the BrOA, we have put effort to understand the dynamics of the carbonate system and CO2 fluxes in different regions of the Atlantic Ocean. And so we are going to talk about some of this region. So let's start. Okay. Let's start talking about the tropical Atlantic Ocean, that is this region here. The Western tropical Atlantic Ocean is a very complex region to understand because of the different surface currents. So we have influence of the North Brazil current and the North Equatorial current and North Equatorial counter current. And here, a very important feature in this region is the high input of fresh water from the Amazon River. So the Amazon River lies approximately here, and they put a lot of fresh water in the Western tropical Atlantic Ocean, driving a lot of difference in the carbonate system and physical properties. The fresh water from the Amazon River develops Amazon River plume that is a very huge area of plume throughout the Western tropical Atlantic Ocean. So the Amazon River plume is The Amazon River plume is a strong CO2 uptake region for the CO2, atmospheric CO2. And this uptake just in the Amazon River plume water correspond to about 87% of the whole CO2 uptake by the entire Western tropical Atlantic Ocean. And another important aspect that we discovered in this study was that water is under the influence of the North Brazil current and the North Equatorial current has experienced an increasing trend in the sea surface PCO2. So the sea surface PCO2 is increasing in a trend higher than the atmospheric PCO2, and this could be very important for the biogeochemistry in this region in the future. But although the importance, the role of the Amazon River plume on the carbonate system is still poorly understood in this region. And this is essentially because of the large gap, both spatially and temporally in this region. So we can see in the time series here, in this figure, in the green time series, that the Amazon River plume is one of the most undersampled region in the Western tropical Atlantic Ocean. Well, this trend in the sea surface PCO2, we can see in another data source, for example, from the BrOA that lies approximately here in the boundary between the North Brazil current and the North Equatorial current. So in this work, in preparation, Musset and collaborators look for reconstructing the carbonate system parameters from the hydrographic data from the BrOA. So what they can see from now is that there is an increase in PCO2 in the last 2 decades followed by a decrease in pH in the same boil. Well, now look for more south in the South Atlantic Ocean in this study. Andrés Pinhango and collaborator looked at the ocean acidification in anthropogenic carbon in the South Atlantic Ocean from the different sections in different years. So what they discovered essentially was that in a zonal section, in meridional sections, was that the western margin in the western margin of the South Atlantic Ocean in the south limit, the anthropogenic carbon uptake is higher than in the eastern boundary, and in the northern limit. And we can see here in this figure that the anthropogenic carbon input in the western portion in the south limit is high as well. So, and about the water column inventory of the anthropogenic carbon, they calculated 0.71 mole of anthropogenic carbon per square meter per year. And this rate of anthropogenic carbon intrusion is higher than the average for the global ocean. So this is very important because this highlighted the importance of the ocean acidification in the South Atlantic Ocean. And in this figure here, in the A subplot, we can see that the difference in the pH, the decrease in the pH is even higher in the central waters than the intermediate water in the deep layers. So most of this increasing in the acidification center water is driven by the submesoscale, by mesoscale adds delivered from the Indian Ocean, the Agulhas adds. So the mesoscale, the Agulhas adds actually is important features in the South Atlantic Ocean because they transported waters from the Indian Ocean throughout this Atlantic Ocean, and they can reach the continental shelf of the South America. So in this paper, FURG used a very large data set for adds in the South Atlantic Ocean, and they discover that both anticyclonic adds from the Agulha system and cyclonic adds from the Benguela system, upwelling system, enhance the CO2 sink into the South Atlantic Ocean. So here in red, we can see the CO2 fluxes associated with the anticyclonic adds, and in the blue, associated with the cyclonic adds. And we can see that both structures uptake more CO2 than the surrounding waters. And the difference between them is that the anticyclonic adds tend to decrease the CO2 uptake throughout their lives, differently from the cyclonic adds that they maintained the CO2 uptake, the strong CO2 uptake throughout their life, in more pronounced than the anticyclonic adds. Well, we are also interested in understanding the role of the phytoplankton and the phytoplankton groups in the controlling of the carbonate system as a whole and in the CO2 uptake. In this study, Andrea Carvalho explored spring and some data set to understand the role of different phytoplankton groups in the CO2 uptake. So what they discovered was the smaller cell size phytoplankton, such as the cyanobacteria, for example, is associated with the oligotrophic condition and leads to CO2, is associated with a CO2 release condition, mainly in the south portion of the continental shelf of the South Brazil and Patagonia continental shelf. On the other hand, larger cells phytoplankton larvae, such as diatoms, are associated with the nutrient-rich conditions, and these phytoplankton groups lead to strong CO2 uptake by the oceans. Another important aspect here is that they describe a meridional gradient of the influence of temperature, change in temperature, and the biological drift on the partial pressure of the CO2 in at this sea surface. So what they discover here is that the influence of the biology activity is higher in the south portion, and the change in temperature plays a larger role in the dynamic of the PCO2 in the north of the continental shelf. But mostly the sampled for this study was carried out in the summer and mostly in the spring. So we actually don't know very well what happened in seasonal cycles in other seasons, and even we know even less about the interlinear variability of this scenario. Well, we can see that in this study was such a review paper, and they, after collaborators, explored all of the available data set for the continental shelf of Brazil. So here in this figure, we have, if I'm not wrong, I think about 100% of the data is from the SOCAT data set that make available PCO2 data. And what they actually emphasize here in this study is that along the continental shelf of Brazil, different drivers, process driver the differences in PCO2. So we have, for example, influence of upwelling system and freshwater input from rivers, and temperature change in some regions, essentially in the south, and currents and coastal complexity. All of these processes influence different regions. In some region, all of them acting together to change the PCO2. Meanwhile, the very important highlight in this paper is that there is a very high spatial and temporal data limitation all of this region. So we can see, for example, in this dispersion diagram, you have more in the X, we have the CO2 fluxes, and positive values indicate the CO2 release from the ocean to the atmosphere, and negative values indicate the uptake of CO2 by the oceans. And we have quite a gradient for the south portion of the continental shelf acting more as a CO2 sink, and the CO2 releasing as we can go further north. Well, another very important region that is very undersampled, not just in Brazil, but in the Southern Hemisphere as a whole, is the estuarine environmentals. So we can see, for example, in this Patos Lagoon estuary is a region restricted to this red rectangle here. And the Patos Lagoon estuary is an estuary zone of the Patos Lagoon that is known as one of the larger coastal lagoons in the world. The Patos Lagoon have been studied from different aspects to understand the dynamic, the water dynamics, and the sedimentary dynamics, and essentially from the physical perspective in the last 5 decades. But this paper was the very first study on the CO2 system in the Pat's Lagoon estuary, even though the importance of this region economically and for fishery and different aspect, not just for economic aspect, but the scientific aspect as well. And in this paper, Cynthia Albuquerque and collaborator describes the seasonal cycle of the CO2 system. And they discovered that the CO2 system swings between the ocean dominated, that is dominated by the intrusion of the seawater, essentially driven by the winds forcing. And these occur essentially during the summer, in the autumn, and behaving as a riverine-dominated region, mainly in the spring, in the winter, when the river discharge is higher. But with actually the water and CO2 exchange, these regions were never addressed. So this is just one example of how estuary environments are undersampled and misunderstood in the South Hemisphere, but definitely in the Brazil, because we have some another examples of some important, very first studied in the northeastern, in the southeastern of Brazil as well. This is another study carried out in the same region, but to understand the daily variability of the pelagic ecosystem metabolism. So what the Borgin and collaborators discovered here was that in a short time scale, in a daily scale actually, and the pelagic ecosystem metabolism characterize a net heterotrophic behavior in the estuary. So we can see here in this figure the variability, the daily variability of the net primary production, respiration, and gross primary production, and the pelagic ecosystem metabolism. But what I would like to highlight here is that the ecosystem metabolism is essentially behavior because of the consumption of the oxygen and the negative values of the pelagic ecosystem metabolism. And they also identified that the water air CO2 uptake, yes, the estuary essentially in the daily time scale uptakes CO2 from the atmosphere, and this is influenced by the diatom carbon accumulation during the summer. Well, let's talk about the Antarctica. Okay, let's talk about the Antarctica. So this study was carried out by it's a very new paper, recent paper from the Mauricio Santos Andrade, and we explored the long-term variability, the decadal variability of the carbonate system in the northern Antarctic Peninsula here. So in this region, there is a strong mixing between CDW, the circumpolar deep water, and the density of water from the Weddell Sea. There are completely different water masses in terms of the physical and biogeochemical aspect. And this mixing between these two water masses is modulated by the SAM and ENSO in this region, mainly in the central basin, as we can see here, by the profiles in the central basin, but the eastern basin is quite homogeneous throughout the water column. And another important aspect that they discovered here was that in the deep layers throughout the northern Antarctic Peninsula, there is a change only in dissolved inorganic carbon associated with no change in the total alkalinity. But we don't know very well why this is happening, because this is characterizing the exchange of CO2 between the ocean and the atmosphere. And this is obviously not happening here because it's at the deep. So this is certainly one of the issues that should be addressed in the future studies. So probably this is because the mesoscale process that is important throughout along the Northern Antarctic Peninsula, as we can see in this study by the mini collaborators this year, they explored what the importance is, what are the role of the mesoscale add in the carbonate system. And what discovered was that the CO2 release is due to the enhanced dissolved inorganic carbon that is modulated by the upwelling intermediated waters and the respiration. So because of this increase in the dissolved inorganic carbon, the waters inside the air in the surface release CO2 to the atmosphere, completely different what happened in the waters around the air. So one of the main things I would like to emphasize here is that the mesoscale process, such as the edge, for example, are important drivers and should not be neglected in the dynamic of the biogeochemistry. But this is just a snapshot for 1 year, 1 season. We don't know the interannual variability and the phytoplankton dynamic, and we don't know very well the net effect of these mesoscale features in the CO2 fluxes and dynamics. So just to summarize, and the take-home message here is that the mesoscale and regional features should not be neglected in the studies, even in large-scale visualizations such as models and climatology. And we know very well the the global behavior of the oceans in terms of the CO2 single source, but we don't know the temporal variability and the magnitudes of these fluxes and the importances of these mesoscale features in a global scales. We have been seeing a pH decrease at about 0.02 units per decade, and the central waters are acidified faster than the surface and intermediate waters in the South Atlantic Ocean. And we do need more observations to improve our understanding of the marine biogeochemistry. So in this table, we have summarized of what of our level of knowledge in different regions in the Atlantic Ocean. And as we can see, we need to improve our understand our samples, our time series to improve our understanding. So thank you very much, and sorry for the time. Yes, if you would like to know more, you can contact me and contact through our social medias and through the email. Thank you.
Thank you, Thiago, for this interesting talk. I will let Kalina to thank also. And then we're gonna have a few minutes to reply some questions.
Yes, thank you, Carla. Yes, I just want to thank the moderator and the speakers and all of you in the audience as well. And if you would like to stay up to date with the GOA-ON community, you can consider signing up as a GOA-ON member. And I can drop the link in the chat. The next webinar that we have scheduled will happen in early August. And one moment, let me pull up the slides here, and it will be by moderated, or it will be presented by the North American Hub and moderated by Dr. Richard Feely. And Li Qing Zhang will present on the global surface ocean acidification indicators from 1750 to 2100. And Dr. Jessica Cross will present on NOAA's recent research strategy of carbon dioxide removal. And so we will be announcing the specific date and time of this webinar soon on our email listserv as well as the website. So I will turn it back to Carla for questions and answers. I know we are going just over the hour, but if anyone would like to stay on, it looks like we've had quite a few interesting questions come through. So once again, thank you to everyone for participating.
Thank you, Karina. So I'm gonna start with the question that Martin Hernandez John asked. He says, congratulations to both speakers. And he asked one question for Michelle and one for Thiago. He has. So Michelle presented that depth in the oxycline is becoming shallower, but there are no data for the last few years. Do you know if it follows the same pattern? And then I'm gonna ask the one for Thiago.
Okay. Well, the last year, as I show, we have a lot of El Niño events. And when we have El Niño events, this change dramatically the depth of the oxygen minimum zone. And for example, this year decreased a lot of the primary productivity. Probably, yes, for that we need to take longer and longer time series, because when we have this change in these short times, change. But the last actualization of the trend in the oxygen indicate that we have a decrease in the oxygen levels, but probably a different pattern between the oceanic. A decrease is is significant in the oceanic part, but in the coastal area, probably because this effect of the Kelvin waves and others, we can have another pattern that can stay, and sometimes we have oxygenation and combine. In the oceanic part, yes, the tendency continue. In the coastal area, we need to observe more.
Thank you, Michelle. I'm gonna continue with the second question for Thiago. So Thiago, does the changes in the mesoscale that you mentioned have repercussions on the CO2 fluxes? But why are there changes?
Okay, so I think this is depends on the region we are talking about, actually, because in Antarctica, for example, the Northern Antarctic Peninsula, we have a very complex system of currents and water masses. So the edges can trap some water masses with some specific characteristics, such as CDW with high CO2 content, and increase the PCO2 and release CO2 from the ocean to the atmosphere. But we can see, on the other hand, in some region, the input of CDW, that is a warm water masses, enhance the meltwater, in some coastal region, that can enhance the biological productivity and behave positive in terms of the CO2 fluxes. But I think in general, it depends specifically the region we are talking about,
Thank you, Thiago. So I see there are some participants that just raised their hand. Maybe, I don't know if Matthews Batista or Cecilia Chapa would like to talk and to ask the question on live. I don't know, it's...
Hi, my question is for Thiago. Well, first of all, congratulations, great talks, both of them. And I was wondering, when you presented the evolution of the eddies and the fluxes, how did you measure the flux along the eddies for such a long time?
Okay, so I think you are talking about the South Atlantic edge, the Agulhas edge. I'm not a co-author in this paper particularly, but I do know the authors. I can share with you if you would like. But I know that we have some tracking measurement of the CO2 underway, you know, with the shipboard, measuring the CO2 fluxes, and some of the partial pressure of CO2 is estimated from the total alkalinity and total inorganic carbon, you know, throughout the cruise track ship, you know.
Okay, thanks. Yeah, I was wondering, like, if you were like following around the eddies or used opportunity ship data that casually cross them, because we have a lot of eddies in here in the Gulf of Tehuantepec in southern Mexico, and it's very interesting to know how they evolve along the time. But yeah, I will appreciate the data if you have the names of the waters. Thanks.
Thanks, Cecilia. Just to finish, there's another anonymous question, and to both, as how can we better integrate mesoscale processes in ocean acidification studies? I think it's a great question to wrap up this talk.
You can start, Michelle.
Okay, well, I think, well, Thiago have more experience, but yes, this is true. The mesoscale is something, well, is very important. I said, for us, for example, to integrate, we need to go to make a deeper research on mesoscale. We start some lines about eddies. For that is, like Cecilia said, it's very interesting, Thiago, the way that you say, well, for us it's more complex, no? To follow, and the opportunity. We need to have the opportunity with ships, but also we have another strategy that could be very interesting is with gliders. Now we have some gliders in our system, and the glider have the opportunity to track, no, with high resolution. And this could be, for example, for us, some strategy that we can implement in order to track the eddies. Then we need to go and for the carbonate system, I think, I don't know if there is some sensor that we can put in the gliders, but it's not, we need to have the opportunity with a ship and combine the ship with autonomous sensor like glider, for example, in order to continue or start this kind of research. I don't know, Thiago, if you have another idea.
Yes, I totally agree with Michelle, actually. So I think the easy answer is that we need more data with more time series and with the different time scales, you know, from daily time scales, monthly, seasonal, interannual, decadal. This is the easy answer. And the more complex answer, I think, that needs one more webinar because it's very difficult, you know, but I think we do need to match the time scales, short time scale with larger time scales, with the method described by Michelle before. Thank you.
And probably, Thiago, the modelers are very important also, no? Combine the observation in situ plus a good physical model in the area in order to understand the patterns and the force in the eddies, for example, and combine with the chemistry models. I think models is a very powerful also method.
Yes, definitely. I agree.
Thank you, everybody. Thank you for your questions, for the time. There was another questions that we didn't have time to reply, but you can contact the panelists directly. The emails are on the screen right now. So I'm gonna pass to Galina to close this webinar. And thank you so much for all that attended, and of course to the presenters.
Yes, and thank you, Carla, for moderating. Again, keep an eye out for future webinar announcements on GOA-ON, and we hope to see you on other webinars. Thank you for joining today, and thank you to the presenters.
Thank you to GOA-ON, to LAOCA, and to all the community. Thank you.
Thank you, everyone.
Thanks. Bye.
Thank you. Bye.